So, Dr. Romero-Olivares, you're about to start a faculty position at University of New Mexico, right?
So New Mexico State
Oh, New
University.
Mexico State. I'm sorry.
Yes, so I'm starting in August
Awesome.
2020. So in the mid,
Yeah.
I'm able to start, but if everything goes well, I will.
Yeah, it's kind of a weird time to be starting a new position, huh?
Yeah, definitely. But we, like fungi, we can adapt too. So we'll be fine.
Yeah, awesome. Yeah, so do you want to talk about fungal adaptation?
Yeah. It's something that I'm very interested in and something that I've looked into in my research. It's I've I've looked at it in a kind of like in a different lens than most traditional evolution studies because my main focus is ecosystem ecology and soil ecology. So it's very difficult to study evolution in a more traditional sense while not being able to see the organism that you're working with in most cases. But
Yeah.
yeah, I've done studies in the lab, doing more like following more of like a traditional experimental approach, and I've also been exploring some other things in the field that aren't necessarily traditional in the evolution context, but may provide us some hints on evolutionary processes in the field.
Cool. Yeah. Do you want to get into it? What were your, because you had a recent publication come out about kind of like adaptation to global warming or experimental warming, but kind of trying to simulate the future a little bit.
Yeah. So one of there's like one big question that we don't know the answer to, and that question is: are fungi adapting to global climate change, like some of the like increases in temperature, or like drought, or like some other environmental stresses that are brought by global climate change. So I guess like in my work during my PhD, I looked at warming as an environmental stressor. In my postdoc, I'm looking at nitrogen pollution as an environmental stressor.
Oh, cool.
And the paper that I published recently was some of my work that I did during my PhD. And one of the questions that we're trying to answer is, how does adaptation in fungi look like? How are they changing in terms of their physiology, and how are they changing in terms of like bigger scale types of responses? Like, how is it going to affect us? And the way that I look into that is by focusing on the carbon cycle. So basically, how much carbon fungi are decomposing, and what portion of that carbon is being returned to the atmosphere as CO2. And in that paper that I published recently, I found by looking at just gene expression of the fungi in the soil, I was able to find out that fungi that are experiencing warming and drying are too stressed. And all of the resources that they're investing, it's to just staying alive. So they're really not investing any resources in decomposing. They're really just investing a lot of resources in staying alive. That on itself doesn't really tell us if fungi are adapted or not, but it's providing some hints about how they may be adapting. So I could just hypothesize that probably fungi, like the fungi that are adapting and those that aren't dying and staying alive, are those that are investing more resources in just metabolic maintenance.
So just to back up super quick, what I know, like there's a lot of different fungi that do a lot of different things, but the fungi you're mostly focusing on, they're just generalized decomposers? They're not like mycorrhizal fungi or?
Yes, exactly. So the fungi that I'm focusing on are saprotrophic fungi. So those that are free living in the soil.
Cool. And so I know for some types of organisms, we're really worried about climate change leading to extinction, but do you think, can fungi go extinct?
That's
Is this a
a
silly question
no,
or?
no, no. This is a great question, and I don't think we know that. We,
Yeah.
I think they totally can, and probably many fungi have gone extinct at
Mhm.
this point, but just we don't know because we don't know the.
Right.
Just like most fungi, we just don't know them. Most of them, like we've never seen, and we only know them based on this like random DNA sequence that we're able to extract from the soil. But yeah,
Mhm.
I think that fungi can get extinct. And yeah, and we don't know which ones have gone extinct.
Yeah, I guess probably the fungi that are super duper specialized with organisms that we know can go extinct might be more likely to go extinct. But
Yeah,
if it's like a really
exactly.
specialized orchid mycorrhizal thing or something, or
Yeah, exactly. Those
um,
that have like just like very
like a species
specialized,
specific,
yeah, species
yeah.
specific host,
But
probably.
for the generalist, yeah, it's just a total black box. We have no idea for
Yeah,
extinction rates or.
yeah, we have no idea. And there are some fungi, like even within like the generalist fungi or
Mhm.
saprotropic fungi, even within those, like there's a spectrum of those that are found worldwide versus some other ones that seem to be found in under certain conditions only. So
Mhm.
even within the I guess like the less specialized fungi, there's still like a good degree of specialization.
Oh, that's really cool. Yeah, I guess I haven't really thought about. I guess I have some kind of mycorrhizae studying friends that, and we've kind of talked about like geographic range stuff with fungi, but I don't know anything about the saprotrophs. Yeah, that's really
Yeah,
cool.
it's kind of relatively new
Yeah.
area of research. I've seen in the past couple of years, I've seen just like new studies looking at like like global distribution of certain fungal species. And
Mm.
there's definitely some species that show global distribution patterns and some others that don't.
Yeah, is California, because a lot of your work is based on California and then like Baja, right? So
Yeah, so I've done work in California, and also in
Mhm.
Alaska.
Oh yeah, Alaska, right? That's so
Yes.
cool. So Alaska, why is Alaska cool to study from a global warming perspective?
So being on the very north of the globe,
Mhm.
global warming is being affected the most. I guess like global warming is affecting the most those ecosystems on like higher latitudes. I mean, it's affecting all the world, but some
Yeah.
of the effects are just like very obvious and somewhat extreme in the northern hemisphere. Um, so that was like the main reason, like let's go to a place
Right.
where we're going to be able to see the effects like fast, and that it's going to inform us on what's going to happen to the rest of the world further down the line.
Yeah. So were you working on like permafrost stuff, or was it
No, I was working
south of there?
in boreal forest, so just like
Okay.
a bit south of the like permafrost soils, but like
Gotcha.
still yeah, so boreal forest, temperate forests, and peatlands and Arctic ecosystems are the ones that are being affected the most.
Hmm. Yeah. So, and I guess just to kind of circle back, you were talking about, we only really know the fungi because of their little DNA sequences.
Yeah.
I know you're working a lot with like kind of environmental DNA sequencing, or like I know at least one of your papers is metatranscriptomics. And
Mm-hmm.
could you talk just a little bit about that so the students,
Yeah.
they don't have to understand everything, but
Yeah.
I'll stop you if you're getting too into the weeds, but it'd be cool
Yeah.
to hear about some of that.
Yeah, I am an expert in just working with environmental samples, particularly soil.
Mhm.
And something that I get from very often is that people just assume that I know a lot about mushrooms because I work with fungi. But
Mhm.
then when I let them know that I actually don't work with mushrooms, that I
You
work
work with
with,
A, T, C, and G.
yes, exactly, that I work with the DNA from
Yeah.
fungi in the soil that you can't see.
Mhm.
It sometimes creates a lot of confusion. But yeah, that's what I do. I basically take a soil sample from anywhere.
Oh no, I think you've frozen for a second.
Can you see me now? Can you
Yeah,
hear
yeah.
me?
I think your internet or someone's internet just
I think
froze up for
it's
a
my
second.
internet. Okay,
Sorry.
I hope it doesn't cause any trouble.
You're good. You're
Okay.
good. But maybe start over from the DNA soup.
Yeah, so what I was saying is that I usually just like get a soil sample and then extract DNA and RNA.
Mhm.
So basically just all the DNA and all the RNA that is in the soil, which, like that can be from fungi, from bacteria, from plants, from insects, from nematodes, just like basically anything that's in the soil I extract.
Yeah.
And then once I have that wealth of information data, like per sample, I usually get like a 1 terabyte of genetic data. And then after that, like all of the like processing of the data is through basically just like programming and a lot of many hours
Yeah.
doing programming and just like computer work and then further statistics. So yeah, but all of the information that I get, it's from soil samples. So
But
not
so how?
mushrooms, soil samples.
Yeah. But so how many species do you, or how many operational taxonomic units do you get from like per sample? What's
Um,
the diversity levels that we're talking about here?
Oh, so many. I mean, from each of my samples, I usually get at least a couple thousand OTUs from each single family of fungi that's described. So, like most of the fungi that we know exist are usually represented in each of the samples that I have.
That's
Plus, a
wild.
ton, the majority, I would say, of the samples are
Yeah.
unknown organisms and unknown fungi that aren't described.
That's wild. And how big are your samples? Is it like a soil core like that or
Oh my
something?
gosh. No, they are so small. So do you know like the Eppendorf tubes, like the
I do.
1.5
Yeah.
mils?
Yeah.
Okay. So it's
I don't
like,
have any right here, but yeah, they're
yeah,
that
so
big. Yeah.
it's not even that. It's like 1/4 of that.
So thousands of species in that much
Yes.
soil. Yeah.
Yes, exactly. Which it's mind blowing.
That's super wild.
Yes.
Um, can we just talk a tiny bit about the advantages, like why you would want to look at DNA, but also check for the RNA from like a ecological standpoint, or for whatever you want to, yeah, say about that?
Yeah. There's so when you extract DNA from your soil samples, you get a sense of who's there.
Mhm.
But you cannot know what are they doing. So you just know like these are the species in this soil sample. Like what are they doing? Who knows? You don't
Yeah.
know because you only got DNA. So that is the power of having RNA, is that you can know what's going on. It gets kind of tricky though, because with RNA, depending on how you process the data, you may know what's going on, but you don't know who is doing it.
Right.
So it can get kind of tricky because I'm an ecologist and an ecosystem ecologist. Um, for the purpose of my research, is somewhat more important to know what are they doing rather than who is doing it. So
Hmm.
that's one of the reasons why I focused in RNA in the past, just to get a sense of what are they doing and how it affects the carbon cycle. If you just look at DNA, you can't really make any assumptions of, this are the organisms here, but what is their relation to the carbon cycle? Who knows? So then that's why I focus on RNA.
Yeah, because RNA it degrades pretty quickly after it's, if you think about the central dogma of biology or whatever, right? That's the
Yes.
intermediate between DNA and the proteins, and RNA doesn't stick around nearly as long, except for ribosomes,
Yes,
as
exactly.
DNA. So you get more of a
Exactly.
sense for like who's active, or like what's actively being done,
Yes,
give or
exactly.
take a few hours sometime, right? Or a
Oh
few
my gosh,
days or
yes.
something.
And even minutes, it could be.
Wow.
So it is very like time sensitive, and it's like RNA, as it's known, it's very unstable because it's a
Hmm.
single chain rather than two.
Yeah.
So yeah, that definitely complicates further some of the issues of working with RNA. One
Yeah.
of the good things is that you can buy solutions that freeze the RNA
Hmm.
when you when you soak your samples in it. There's some, arguments about that being effective or not, but that's the best thing that we have right now.
Do you collect into RNA later, or do you do like nitrogen freezing or?
For my work in Alaska, I did RNA later because it's it's a really remote site, and I couldn't possibly carry liquid nitrogen with me. But in the place that I'm working in right now, it's at Harbor Forest, which is really close relatively to the university where I am right now in New Hampshire, and it's totally feasible to carry liquid nitrogen with us.
Oh, that's really nice. I've done a tiny bit of work at Harbor Forest, and that's like not a bad place. It's
so great. I'm so grateful that we have that. It's such a great investment of land, I think.
Yeah, it's kind of like the Drosophila of northeastern forests, because it's like so many people have studied, different aspects of that landscape, and so
it's really wonderful, and it's like so much great science has come out of Harbor Forest. It's really amazing.
Yeah. Oh, that's really cool. I feel like there's something else I wanted to ask you about the meta-transcriptomics, but I'm drawing a blank. Are you excited about, do you have any kind of upcoming projects that you're kind of like thinking about that you're, that you can share without like blowing any secrets or anything? Or
yeah, definitely.
This is like a talk show with like, like no spoilers,
Yeah. But, I'm still very interested in, I guess, metabolism just in general. So again, looking into function and what's going on at the ecosystem scale. And for the past few months, I've been exploring volatile organic compounds. So those are just secondary metabolites produced by, basically every single living organism.
A lot of things. Yeah, I guess I don't think of fungal VOCs. I haven't heard much about that, but that's
oh
cool.
My gosh, there are so many.
It's a thing, huh?
It's a thing. It's relatively new as well, mostly because some of the machines that we have now to actually look at VOCs are better, but they were really not great for a really long time. But it's getting better now, so it's like an emerging area of research. Now we have the tools to actually look into them, and I am very interested, looking at VOCs again in the context of climate change. So I'm really one of the things that I found in my meta-transcriptome paper is that there was a lot of activation of secondary metabolism in some of the fungi. So I think that many of the fungi that are trying to survive may be doing so by investing on their secondary metabolism. So probably investing some resources in producing certain, just like antibiotics or certain VOCs that may be securing their sort of space and resources.
So it's like an antibiosis kind of thing for?
Yes, definitely. So
okay.
So some of the VOCs are most of the VOCs that have been described in fungi have some sort of antimicrobial function, which makes me think that they're probably producing those secondary metabolites to actually inhibit the growth of bacteria or of some other fungi. So that's what I'm looking at right now, just exploring how, again, warming and nitrogen pollution is affecting the emission of secondary metabolism in fungi. So that's one of the interests that I have. And the other interest is that some VOCs
I think we might have lost you again for a second.
But I'm interested in looking at from a fungal perspective.
I'm sorry. I might have to ask you to.
When I'm back, let me know.
Yeah, I think you're still frozen. Oh no. Yeah, I don't know if I can hear you pretty okay.
Not yet. But you can.
Yeah, your picture is frozen. I don't know how to stop that.
should I off and then?
Oh, maybe could you turn your video on and off or something? Or yeah, you could just turn your video off and we could see if we can hear you.
Okay, video off, and then if I get on again,
Oh, this is a bummer. Yeah, I don't know. Yeah, even your
no,
sound is a little bit hard to understand. Oh,
no.
you're back now. You seem to be
Sorry.
moving. Oh, okay. Yeah, you're in a new...
I'm sorry. And now you saw my husband. I was in a room, which is kind of like far away. Can you see me now?
Your picture's moving. It's not like super smooth, but I think it's a lot better, and your audio is better.
Okay, so better now.
Awesome. Yeah.
Yes.
That sounds great.
Okay.
Yeah. I'm sorry. Can I ask you to rewind a little bit the VOC stuff?
Yeah. So I think probably the last thing that I said was that I'm not only interested in seeing how VOCs affect other fungi or other microbes, but there's a lot of VOCs, and this has been explored in plants, but not really in microbes, is that some of those VOCs are precursors of greenhouse gases. So some of those VOCs are just being emitted naturally by organisms, and some of those VOCs, when they go into the atmosphere and react with whatever's in the atmosphere, like hydrogen, oxygen, and so on, they turn into greenhouse gases.
Oh, wow. So that's potentially bad. So stressed fungi might emit more VOCs that are potentially...
That's what
gases.
that's what I'm starting to explore. It's like still very, new and have no what's the word, evidence, I guess, to suggest any of this, but that's what I'm interested in exploring.
Worth checking into, though, for sure. Because that's, I mean, it's a big group of organisms, and they're all over the place. And
yeah, definitely, which is one of the main reasons why I'm interested in them.
That's really cool. Do you know anything? Are you going to be working in New Mexico? Like, are you going to have field sites there, do you think? Or
I am. So for the first couple of years, I'm going to continue to work in Harbor Forest, just because I got started here and the project is ongoing. But I'm a huge fan of LTRs. So for those of you who don't know what that is, it's like the long-term ecological research stations. Harbor Forest is an LTR. And New Mexico has a few LTRs around, and I'm especially interested in working in one that is called La Jornada. And within La Jornada, there's disturbance LTR, which I'm very interested in looking into.
Oh, cool. So there was disturbance before the LTR was set up, or there's like a gradient?
So like exactly. So the LTR, like the researchers in charge of the LTR are creating the disturbance.
Oh, cool.
Yeah. So basically, they just go with like machines that have like some sort of, I don't know, like some sort of like metal holes, and they just like walk around with those machines on the LTR and create disturbance.
Oh, that's so funny.
Yeah,
Definitely. That sounds like a fun job.
Right.
Yeah. Cool. Well, I don't want to like keep you for, forever. It's really fun hearing about your research, though. So, I mean, thanks so much for stopping by and
oh my gosh,
taking the time to talk.
No, thank you so much for inviting me. And if anyone has any questions, I am happy to be in touch with anyone who's interested in fungal adaptation, and you can give them my contact info.
Yeah, I for sure will. Yeah, okay. Yeah, well, thanks so much, and great to meet you, and stay safe.
Yeah, you too. Thank you so much, and sorry about the internet inconsistencies.
Oh yeah, no worries. Yeah, thanks a lot.
Thank you so much. Yeah,
see you.